Okay.
Sure, he called me.
All right.
Turning you in, Jay. Bringing down my self-esteem.
All right, guys. Quiet. Okay. Today, we are beginning the most important conversation about a process called meiosis. Okay. Now, meiosis. We've already talked in great detail about mitosis. Remember, mitosis is the process where genetic material is divided up. That's mitosis. Now, in mitosis, that genetic material was divided up equally. So if you started off with 8 chromosomes, you're going to end with another cell with 8 chromosomes. And also in mitosis, we've already talked that genetic material is going to be identical to the parent cell it came from. So you're making a twin. Mitosis has a T in it. You're making a twin cell. You're making a clone, basically. So we already talked about those details. And remember, this was a process that took place in 4 phases. Remember PMAT, P-M-A-T, prophase, metaphase, anaphase, telophase. The good news is in meiosis, which was what we'll talk about, those same steps actually occur again. However, you're going to see they happen twice, each of them. So you go through a PMAT once, then a cytokinesis, then another PMAT, and a cytokinesis. So it's like takes twice as long, basically. There are some other differences in meiosis, but we'll get to those details in a minute. But before I get to that, you need to know a couple of terms, and they're pretty easy. You need to know the difference between haploid and diploid. So 2 vocab words. Actually, they are vocab words from what I understand. Okay. The technical definition of haploid is referring to one set of chromosomes. The definition of diploid, 2 sets of chromosomes. Okay, well, here, not done yet. If a cell is haploid, it has only one set of chromosomes. They give that a symbol to represent, little n. If a cell has 2 sets of chromosomes, they give it a symbol, 2n. This is kind of easy to remember. The word di, the prefix di, di, means 2. Okay, so, and then I just always remember haploid's the other one. So haploid, 1 set, diploid, di, 2 sets. Humans have how many chromosomes in each of our cells?
46.
We have 46 chromosomes. Okay. That's our diploid number. So, for example, humans, our diploid number, our 2n number is 46. Okay. What would a human's haploid number be? 23. Heard it from someone. Someone else tell me what type of cells that humans have 23 chromosomes. What's that fancy word? Or give me an example, or just any cell. What kind of cell do we have that has only 23 chromosomes? Did I hear it?
What?
What type of cell in the human body has 23 chromosomes? One set of chromosomes. Gametes, which are either eggs or sperm, depending on if you're male or female. Gametes are haploid. Okay. All the other cells in our body are diploid. We give those a term called somatic cells. Somatic cells, which is just a general name for any cell in an organism other than your sex cells, are going to be diploid. Okay. So, gametes, egg and sperm. Let me draw like a little sperm here. Let me draw an egg here. When these are both haploid cells, when the 2 haploid cells get together, they're going to produce something called a zygote. And a zygote is a fertilized egg, and a zygote is a diploid cell. The egg was haploid, the sperm was haploid. They got together, combined to a zygote during the process of fertilization. That's a diploid cell. So we need to get really comfortable with these terms because they're going to come into play a lot here. Okay, well, let's go back to mitosis. I know I said, we're not done with mitosis yet. We've already learned that process. Remember, mitosis makes twins. They're making a clone. In mitosis, you should understand what I mean then when you go from a 2N cell to, I should make another cell here. In the process of mitosis, remember, a cell is dividing into another cell. Each of those cells is identical to the parent, both in number and genetic information. It goes from 2N to 2N. Mitosis. So 2N to 2N. Meiosis is very different. You can't represent it the same way. In meiosis, you're taking a cell which starts off as 2N. I'm going to write a little different way. A cell that starts off with 2N, but after it's all said and done in the process of meiosis, you're actually not going to end up with just 2 cells. You're going to end up with 4 cells. Oops, I'm running out of room. Each of those cells has an N number of chromosomes. In other words, the process of meiosis, you are making new cells. You're dividing up the genetic material, in other words, to go on to produce new cells. But those new cells are only going to have half the amount of genetic information as the parent. That's why you go from 2N to N. And you're going to make 4 of them. Remember, in mitosis, we only went from 1 cell and made 2 cells. In meiosis, we're going to go from 1 cell and make 4 cells. In other words, males have special cells in their body, whether you're a plant, animal, whatever, that undergo meiosis to produce sperm. That process of meiosis. Ladies and females have specialized cells in your body that undergo meiosis to make your eggs. And that's kind of a nice overview. Okay. Moving along to the details. The details, because it's all about the details. Let's go talk about meiosis. Okay, meiosis is broken down, like I said, into 2 parts, and they named them nicely, meiosis 1 and meiosis 2. So let's talk with meiosis 1. Meiosis 1 is going to consist of 4 phases. Prophase 1, metaphase 1, anaphase 1, and telophase 1. They're not too creative when they come up with these. These are words you've already seen before. Prophase, metaphase, anaphase, telophase. And you're going to see there is a lot of similarity between these words here in meiosis and mitosis, but there are very key differences, which I'll get to in a minute. Before I do that, the cell will then undergo cytokinesis. You guys know what that is as well. Remember, that's actually a cell breaking into 2 cells. So if you start off with 1 cell in the beginning, at the end of meiosis 1, down here, you will have 2 cells. Okay, because they'll actually separate by cytokinesis. Okay, but you're not done yet. Then those cells are going to enter meiosis 2. And meiosis 2, conveniently enough, is made of the same named phases, but in their part 2. Prophase 2, metaphase 2. You get the idea. Anaphase 2 and telophase 2. Okay. And then at the very end of this, yes, you will have another cytokinesis. So you can kind of see, you have 2 sets of cytokinesis. You start off with 1 cell. You can kind of see where you can get 4 cells, because each cell is going to undergo another set of cytokinesis, and you're going to get 4 cells. So that's where the 4 cells come from. Literally, you have 2 sets of divisions of cells. Okay, let's talk a little more detail here. All right, you're starting off in meiosis, like I said, with a diploid cell, a cell with a full set, 2 sets of chromosomes. At the end of meiosis 1, the first phase, believe it or not, you've actually already halved the number of chromosomes. You'd think you'd be done, but you're not. Those cells are going to undergo another division to produce N cells. I'll be honest, the nice thing Meiosis II, and I'll write it right here, the whole process is very, very similar to mitosis in steps. In other words, prophase 2, metaphase 2, anaphase 2, and telophase 2 are almost identical to mitosis that we've already talked about. You'll see pictures in a minute. However, there are lots of differences in meiosis 1 that we have not seen yet. Namely, two major things. One of the biggest things is in prophase 1. You have something going on in prophase 1 that we have not seen before, a process called crossing over. Okay, crossing over occurs in prophase 1 of meiosis 1. That is new. We've never talked about this yet. Crossing over in layman's terms, real easy, that just basically means two chromosomes are going to give each other a hug and exchange genes back and forth. That's all that means. We didn't have that going on in mitosis. We didn't want to have genes being scrambled all over the place. We wanted to make twin cells. We didn't want genes to scramble. However, it's very important for crossing over to occur in meiosis, because we don't want the eggs and the sperm that are going to be made as a result of meiosis to look like the parent. Otherwise, every one of you guys in this room would look like your parent. Okay? But you don't. And one of the reasons is because crossing over takes place. It's going to scramble up all the genes on the chromosomes that come from your parents. Okay. Another extremely important step that occurs is at metaphase 1. Metaphase 1, remember meta means middle. The chromosomes are going to line up in the middle. They're still going to do that. But in mitosis, we saw the chromosomes lining up single file, like kindergartners going into school. Okay, they lined up single file. They're not going to line up single file in meiosis 1. They actually line up in pairs. So I guess that's like, if the kindergartners were matched up with a boy-girl, think about it that way. They're going to pair up. They're going to pair up. Remember, we brought up this vocabulary word before, but how are they going to pair up? They are going to pair up. There's a chromosome. There's a chromosome. They are going to pair up with their homologous chromosome. Homologous chromosome. That's the term. That's the fancy term given to two chromosomes that are basically paired up with each other. And again, we've talked about this as well, that those two chromosomes pair up with each other, not by random choice, but by the fact that one of those chromosomes we received from mom way back when and one received from dad way back when. So that's why they line up based on that. Okay, very good. That's it. That's kind of an overview of meiosis. Let's show you some pictures. And it might make a lot more sense. Let me start with this whole transparency I'm about to show you is just meiosis 1. This is just the first stage, the first half of meiosis. Meiosis part 1. Okay, we should all remember what interphase is. So you're starting off with a certain cell in interphase. Remember, that was the crust of the pizza. There's the cells just doing its thing. You can't even see the chromosomes yet in this phase. However, meiosis 1 will begin at prophase 1. And now you can actually visually see the chromosomes. Back then they were just thread-like. Now they've actually condensed to the chromosomes. In this picture you see 4 chromosomes. One there, one there, one there, and one there. Okay. Remember, prophase 1, something very important called crossing over occurs. Literally, what happens is these two are homologous chromosomes. Okay, that's their pair. They are going to kind of cross over and hook up to each other. And where they overlap is where genes are going to move from one to the other. So you can even see from this picture, they're going to overlap. Genes will be exchanged. And you can see from the color shading difference, you have a different combinations of genes on both chromosomes. One started off all red, one started off all yellow. Crossing over occurred. You see little pieces of yellow and red being exchanged between the chromosomes. That's crossing over. Now remember, on a chromosome, there's lots of genes. Oh, tons of genes. Not every gene's going to get transferred over, just some of them. But it's going to basically mix up the chromosome. Okay, that's important. Metaphase 1, they're still going to line up at the middle. This picture isn't super clear, but you can kind of have to imagine this being the middle of the cell. And remember I said in metaphase 1, they're not going to line up single file. They're lining up with their buddy. That is a huge difference between metaphase 1 of meiosis and metaphase of mitosis. They lined up single file. All right, so they're lining up with the buddy. Anaphase, they'll still pull away from each other. Please note, though, in anaphase 1 of meiosis 1, the homologous chromosomes separate from each other. That's going to be different for anaphase 2, but mental note. In this anaphase 1, the homologous chromosomes, the pairs, are going to separate from each other. And then at the very bottom, you have kind of telophase and cytokinesis kind of happening where eventually this cell will become 2. Now, but notice, you started off with a cell with 4 chromosomes, 1, 2, 3, 4. Even though they're X's, it's still considered 1 chromosome. You started off with a cell with 4 chromosomes. Check it out. I'm halfway through the process, and I have each cell now with 2 chromosomes. 1, 2 here, 1, 2 here. But still, they're different genetic combinations. All right, I'm just kind of curious to see if anybody can answer this question. This cell is now going to go through each of these cells is now going to go through a whole other step. What do you think is going to happen in the second step if it's similar to mitosis? What's going to break apart? Anybody know? You should know. Oops. All right, metaphase 2, or meiosis 2. Meiosis 2, at the very top, we are starting where we ended over here. Each cell has 2 chromosomes and they look like X's. Prophase 2, nothing exciting. You do not have crossing over taking place in this step, though. We've already done that in prophase 1. Prophase 2, no. Metaphase 2, they are going to line up in the middle, but look, they're lining up single file now. One there, one there. This looks exactly like mitosis. They lined up single file. Each cell, remember, has 2 chromosomes. 1, 2, 1, 2. Here's what happened. This is the difference. Check out anaphase 2. You can see that in anaphase 2, I just asked you what's gonna separate. It's really important for you to see in this picture that what's going to separate are the sister chromatids. We've already learned that term, sister chromatids. So what's going to separate the X's are going to go their separate ways. It was an X. One half of the X is going to go one way. One half of the X is going to go the other way. Same with both chromosomes. You still have 2 chromosomes in this cell. Even when this happens and you produce cells that look like this is still considered 2 chromosomes. Still considered only 2 chromosomes. But check it out. We're at the very end, 4 haploid daughter cells. Not only have you made twice as many cells, but all 4 of those are haploid. They only have a half a set. They don't say it in this picture, but these are either going to be sperm or they're going to be eggs. Okay. Humans. Okay. Humans like I said, we have special cells in us that undergo meiosis and produce our haploid gametes. It makes sense. Because what would happen if we didn't have that happen? What if our sperm did contain both sets? What if our sperm and our eggs contained 46 chromosomes? Think about what would happen when fertilization occurs. An egg and a sperm hook up and they each have 46 chromosomes. They're going to make an individual with how many chromosomes? 96. Willing to place money on the fact that thing wouldn't be human. All living organisms vary in their number of chromosomes that they have for the most part. Yeah, I mean, humans are very specific. We have 46 chromosomes. We better find a way to make more offspring, and they only have 46 chromosomes too. We don't want a kid with 96 chromosomes. Number one, it probably wouldn't develop, and number two, it definitely wouldn't be human if it did. Okay, so we got to find a way. That's what meiosis and cytokinesis, and that's what this process is all about, is generating that we have to half the number so that they can combine again. But it is so important for you to know these new gametes that are made do not look like the parent in any way, shape, or form. Not even the fact that they have half the number of chromosomes, but because the genes are all randomly disassembled. Okay, so that's actually really important. Again, this is why we don't look like our parents. But you can see it's the same information is there, but if it's scrambled, it might not read the same. So, cool.
Is that it?
I'm kind of checking it over. That's it. I'm not done with today. This is what I want you guys to do. You guys have an activity for me.
I think you're going to give me fingernail polish.
You have, and I'm making all my classes do this, so I'm not singling you guys out like you think I am.
Think.
All right, here's what I want you guys to do, and I do not care if you work. No, I actually keep that rolling for a second because there's people who have not been here this week and they have to do it too. Okay, so here's the assignment for all my biology classes, whether it's basic or advanced. I don't care. This is what all of them are doing. They are to take a cell. I'll get you started here. And I'm going to draw a nucleus. Because you guys all know the chromosomes are in your nucleus. Yeah, you'll eventually have to. I want you to start off with 6 chromosomes. And I'm actually going to make them different colors. I'm going to make 2 of them blue in my case. I'm going to make 2 of them red, and I'm just scattering them anywhere. And I'm going to make 2 of them green. But there's still just 6 chromosomes, okay? The colors will make sense here in a minute. I've got a cell here with 6 chromosomes. It's obviously not human, but if I had you guys do this with 46 chromosomes, it would take you a lot longer. So I have a cell here with 6 chromosomes. You are going to take this cell through mitosis. All the steps. Okay, so let me give you a hint. This is the cell, what it would look like in the G1 interphase. We talked about that. What I want you guys to do is your first step will be to take that cell and tell me what it's going to look like at the S phase. It's going to look different. You better draw me a picture that shows me how that cell is going to look different in the S phase. You might have to go back to your notes. Then I want you to take that cell. I'm not going to do it for you, but but now you're going to take that cell, and now you're going to start the process of mitosis. So realizing I'm skipping a couple of steps in between, your next step, I want you to be prophase. So that's a little head start for you. Yes, you are going to need colored pencils, which I have here in the room, because when you take this cell through mitosis, you're going to need 3 colored pencils. I don't care what color they are. Why would I have you color 2 of them blue, 2 of them red, and 2 of them green? Why did I do that?
Because they're all not the same.
You're right, they're not the same, but if you have 6 chromosomes, you're going to have 3 pairs of them. Just like we have 46 chromosomes, but we have 23 pairs of them. So it makes sense that 2 of our chromosomes are very similar. That's why I colored them this way. Now I'll give you the hint. For mitosis, the colors do not matter. You're like, well, then why are you having us color them different if it doesn't matter for mitosis? Because I want you to still understand that they could come in pairs. Even though in the process of mitosis, it's not going to matter. So that's the first thing you're going to do. Now, Your next thing you're going to do is take that same cell. This is a whole separate sheet of paper. And you're going to put meiosis at the top. And you're going to take that same cell with a nucleus. And I'll even go as far as to make it trying to look completely identical here. I'm going to take that same cell with 6 chromosomes. So that you can consider this like a separate activity. Now you are going to take that cell through the process of meiosis. This is the G1 phase. I'll give you a big hint. The S phase will look the same as what you did for mitosis. Okay, I won't do it, but there you go. Then, just like you did before, it will go into prophase. But now since we're talking meiosis, you're going to take it, it's going to enter prophase 1. This is where the differences will begin. This is going to take like a day or two to do this project. Okay, this is not a quick project, but it's a very, very important project. I told the other class, I go, it seems like it's busy work, but this is probably the best busy work you'll ever do to truly understand the process of mitosis and to truly understand the process of meiosis, is to draw it step by step. Okay, what you'll need to do, by the way, for the record, you will need to do some labeling as well. You need to label what the sister chromatids are. You need to label what the homologous chromosomes are, not on every picture, just once. You will need to label when crossing over takes place. And you do not necessarily need to worry about the mitotic spindle, or you don't really need to worry about, what else is it? Now that one, a lot of people seem to want to draw the mitotic spindle, and I'm more concerned for this activity that you know what's going on with the chromosomes, not the other stuff that's going on. That's what this is more about, the chromosomes. Your book has some extraordinary pictures. In fact, your book has the same pictures I showed you in class here, just here today. These ones, that's in your book. So use your book as a guide. However, I will tell you, these cells only have 4 chromosomes. Oops, there's that one sheet. These cells only have 4 chromosomes. 1, 2, 3, 4. And I'm having you start with 6 chromosomes, so it's going to be different. But use this as a guide, definitely. I want you to show crossing over. Another tidbit of information, when you draw the cell going through meiosis, listen, this is important, you'll need 6 colored pencils to do that properly. You'll need 3 colored pencils for mitosis. You'll need 6 colored pencils for meiosis. Trust me. It'll work out to your advantage in meiosis if you, even though you're using 6 colored pencils, if maybe 2 of them are a different shade of blue, 2 of them are a different shade of red, and maybe 2 of them are a different shade of some other 2 colors. When you start working through these pictures, it'll make more sense why I have you set them up that way. So you can really use any colors you want, but it'll make more sense if you kind of have them like versions of each other, like light blue, dark blue. Again. All right, guys, that should keep you definitely busy between now and Monday. We'll probably finish it up, because tomorrow I will give you guys a lab. And I think that's it.
Come on, you should.